Okay. Good afternoon, everyone. Yeah, so today we are going to look at a topic which is called physiology of test or gustation. So last time when we met, we were looking at what we call olfaction or physiology of smell. So today I'm proud to give you another topic, which is known as test. So basically, we are going to look at the anatomy, and then we're going to go into the physiology, and then after that, we are going to look at the abnormalities or the pathophysiology that comes about when you look at this topic. So going to start first, we're talking about or looking at the anatomy of test buds. Okay, so as you know that test buds, these are anatomical structures that are designed to sense or to detect changes in test. Okay, so as you go on, you're going to realize that you're going to talk about there are about 5 different tests, basic tests, and we'll go through that bit by bit. But now allow me to just break down the anatomy of test buds, which contain receptors that are able to perceive or to detect changes in test. And as you all know, as you learned when you are in high school, we're doing a secondary school, that the test buds are located or they're found in the tongue, but you're going to realize that they extend more than the tongue. So we'll be able to look at that as you go further. Let's go on the next slide. Okay. Now, introduction to test buds or to gustation. So sense organs for test are known as, sense organs for test or gustation sensation are known as the test buds, like I said earlier. So these are designed to pick up the changes in the environment. As you know that these test buds contain what we call receptors. And these receptors detect a change in an environment, okay? So if you look at the basic definition of a receptor, it is defined as a substance, an object, or something that is able to detect the change in an environment. So basically, that change in this environment is test. Okay, these chemicals that you ingest in your mouth, they're able to cause a change, and that change is going to be picked up by the receptors that are found on the test bud, and as transduction will take place. Now let's go. Let's move forward. So test buds are ovoid bodies with a diameter of about 5 micrometer to 70 micrometer, which means that they're very small. In adults, there are about 10,000 test buds are present, and the number is more in children. So no wonder you notice that when you give a lemon to a child and you give a lemon to an adult, you are going to sense that a child will be more responsive or is going to respond more to the lemon as compared to an adult. No wonder you find that even those guys that take a lot of alcohol, like a lot of spirits, you find that their taste buds are a bit more like numb. They cannot be able to pick up very sensitive taste because beer is bitter. Okay, so you're going to find that if you give them a lemon, the way they're going to respond to a lemon is different from the person who doesn't take alcohol. So that has to do with the number of taste buds that can be able to sense and send an action potential or cause what we call transduction. No wonder they're trying to emphasize on a point that in children, they have got more taste buds as compared to adults. That's what the third bullet. In old age, many taste buds degenerate and the taste sensitivity decreases. So as you know, most of the things that are more prominent in young people, they degenerate as a person becomes older, okay? So you find that there's more like a parabola, more like a curve, okay? There's a normal distribution curve. You start from a baby, you start gaining in terms of sensitivity. When you get older, it starts going down, okay? So that's just the nature of life. So adults are less sensitive as compared to babies. Children old people that are worse because they lose most of their sensitivity as they age. All right, continuation. So now, situation of test buds. Where are these test buds located? Okay, so most of the test buds are present on the papillae of the tongue. That's where they're found. You're going to see an image that's going to come after these few slides. So test buds are also situated in the mucosa of the epiglottis, palate, pharynx, and the proximal part of the esophagus. Remember, in the preamble in the beginning, I did mention about where these test buds are found. Like in high school, I just said the test buds are only found on the tongue, okay? But you have been proven wrong that they can also be found on the epiglottis, the palate, the pharynx, and the proximal part of the esophagus. But the ones that we are focused on learning about, the one which are more prominent in detecting the test, we're going to discuss the tongue, okay? So now, types of papillae located on the tongue, okay? So basically, we have got about 3 types, okay? There's what we call the filiform papillae, there's what we call the fungiform papillae, and there's what we call the circumvalent papillae. So these are arranged in different structures. Others are V-shaped, others have got different structures. So this consists of the shape of how the taste buds are arranged on the tongue. No wonder they have got these names, the filiform papillae, fungiform papillae, and the circumvalent papillae, okay? Based on how they are arranged. So here's a diagram trying to show what you have discussing from the previous slide. So you can be able to see that, number one, we're able to see that we have got what we call filiform papillae, okay? There's also what we call the fungiform papillae. So now the filiform papillae, as the text is saying, small and conical shaped papillae, they are situated over the dorsum of the tongue. They contain less number of taste buds, only a few. They only contain a few taste buds, okay? So among this diagram, as you can see here, this is the whole entire tongue, okay? So you can see that we have got also nerve endings or nerve fibers that are able to connect the tongue and be able to transmit electric signals to areas of the brain that can be able to interpret or detect, to interpret the information. So I'm going to find that there's what we call the glossopharyngeal nerve, there's the caudal nerve, and you're going to find that there's also what we call the sulcus terminalis. So this basically simply means it forms more like a V-shaped, okay? Yeah, as you can see there. And you can see that there are taste buds which are found there, okay? So these are different areas where, these are different types of nerves that are able to detect the changes in your mouth, okay? So now, these are the 3 types of papillae. You are going to find that there's what we call the circumvalent, the filiform, and the fungiform. So here, as you can see, there's what we call the filiform. As I said, they say they are found on the dorsum of the tongue, and you can see here that there are only a few, and this is how they are shaped. And this is the area where they are picked from, okay? Then the second one, as you can see, is called the fungiform papillae, okay? So they are round in shape and are spread over the anterior surface of the tongue near the tip, okay? The one people like using for kissing. That's where they are found, okay? Each papillae contains moderate number of taste buds, about 10, okay? So this is an image showing about the fungiform papillae. So if you can see at the tip there, that's where I have about 10 taste buds, okay? Then the last one is known as the circumvalent. The circumvalent, as you can see, it's found in a V-shaped structure along the sulcus terminalis. So now, the circumvalent papillae are the large structures present on the posterior part of the tongue and are many in number, okay? These papillae are arranged in the shape of a V. Each papillae contains many taste buds, approximately about 100 taste buds, okay? So that's what it's showing. So please go through this in your own free time, look at the anatomy, look at anatomy books, try to understand how they are found and how they're organized and how they connect to the nerve endings or to the nerve fibers, such as the glossopharyngeal nerve and the caudotempinal nerve. So if you can see on the images, the way they are arranged and how they are innervating with the test buds, it can show you which one is found at the anterior and which one is found at the posterior. This will be very important when you look at a cascade of events that occur. Now, test buds located in the papillae of the human tongue. Okay, so now test buds on the anterior 2/3 of the tongue are innervated by the caudotempinal branch of the facial nerve. Okay, it's just a branch of the facial nerve. The caudotempinal is a branch of the facial nerve. Those on the posterior third of the tongue are innervated by the lingual branch of what we call the glossopharyngeal nerve. Don't forget that, it might come in MCQs. So now, there's three major types of papillae, like we mentioned, you already know them. Subvalent, foliate, and fungiform are located on the specific part of the tongue. Please don't forget, go through the text, the ones that are found on the tip, the ones that are found at the posterior part, be able to know them and be able to appreciate the information. Structure of the test bud. Test bud is a band of test receptor cells with supporting embedded in the epithelial covering of the papilla. Okay, so now the test bud is a band of test receptors, like I said. Okay, so it's made up of a bundle or a bunch of test receptor cells with supporting cells embedded in the epithelial covering of the papilla. Each test bud contains about 40 cells, which are modified epithelial cells. Cells of the test bud are divided into 4 groups. Okay, we're going to find this type 1, type 2, and type 1, type 2, type 3, and type 4. In the next, I think PowerPoint, the next slide, you'll be able to depict it. They're going to show you how these cells are arranged. Okay, so it's a bundle of receptor cells. So what you're seeing here is types of cells in the test bud. So there's type 1, type 2, type 3, and type 4. Okay, and we have got on the opening there on the tongue, on top of the tongue, there's an opening that goes into the test bud. It's known as the test hole. Okay, so you're going to find that this is an opening. So inside there, you're going to find the one that are just adjacent to the opening of the test hole are what we call type 1 cells. So these type 1 cells and the type 4 cells, they are known as the substancular cells or the supporting cells. Their main job is for support. After that, you're going to have what we call type 2 cells. As for now, that's an area of research that you guys as medical personnel can go and research in. You're going to find that at type 1, these are cells that are, their function have not yet been identified. So it's an area that's open for research. So when you go to type 3 cells, these are the culprits. So if you can see here, it's even saying what? Receptor. Meaning these are cells that are responsible for detecting the change to a stimulus. Meaning when you and I consume something that is sweet, these cells can be able to pick up that. When you and I consume something that is sour, bitter, these are the culprits that are able to detect the change. Okay, this is another image just trying to show you. So if you can see on the other side here, you can see that it's showing one test bud being zoomed in. Okay, as you can see, it's been zoomed in. Yeah, it has been zoomed. So you're going to find that the test pole here is showing that a film of saliva. Then this test pole has got a test bud there. There's a epithelial cell, test cell, basal cell, and there's what we call gustatory afferent nerve. Okay, and there's also what we call the sensory ganglion. So this image is just showing a branch. So these are grooves inside, which show areas where you can find tests. Yeah, like we said, circumvalent, there is filiform, then there's fungiform, etc. So that's where you're finding these test buds. And these are the ones that are responsible for detecting the change to stimulus. Let's go to pathways. So now receptors. The receptor of the taste sensation are the type 3 cells of the taste buds. The type 3 are the ones which are responsible for sensation of taste. If taste bud is innervated by about 50 sensory nerve fibers, and each nerve fiber supplies at least 5 taste buds through its terminalis. So on this diagram, it's showing the cascade or the pathway in which these messages are able to move from the tongue up to the eye centers of the brain, which can be able to detect the changes that have taken. Okay, so now there's what we call, let me just go on the diagram, it's much easier for me. So now, when you go here, you're going to find that there's what we call the first order neurons. They are the ones which are able to pick up the sensation from the taste buds, or they're the ones that are able to pick up the messages, all right? Then there is where we have a synapse between the first and the second order neurons. This synapse is known as, the synapse is formed in the nucleus of the tractus solitarius. That's where the synapse is formed, okay? We're going to find there's what we call a geniculate ganglion, peroxisol ganglion, all these, you're going to find them there. So as you move, you're going to find from the first, the second order neuron, okay? The medial lemniscus, where you have the second order neurons, you're going to find that this is a bundle, a bunch of the second order neurons. So this synapse is going to form in the thalamus, okay? And they are going to form in a nucleus called the ventral-posterior medial nucleus, as you can see there, okay? Which is going to synapse with the third order neurons, okay? So in order for information to come from the tongue up to the gustatory cortex, or the ones that sense the different perceptions of taste, it takes about 3 different types of order of neurons. That's how they are able to move. So here I will read this, which is able to speak whatever that's explained. So the first order neuron of taste pathway are in the nuclei of the 3 different cranial nerves, okay, situated in the medulla oblongata. The dendrites of the neurons are treated to the taste buds. After rising from the taste buds, the fibers reach the cranial nerve nuclei by running along the following nerves. There's what we call the caudotapiny nerve fibers of the facial nerve, like we said earlier, which run from the anterior 2/3 of the tongue. The second one is known as the glossopharyngeal nerve fiber, which run from the posterior 1/3 of the tongue, like we said. The third one is a vagal fiber, which run from the taste buds in the other region, sorry. As you can be able to remember, we talked about the palate, we talked about the pharynx, the proximal part of the esophagus, etc. So all those are covered by the vagal fibers. Axons from the first order neurons in the nuclei of these nerves run together in the medulla oblongata and terminate in the nucleus of the tractus solitarius. Okay, like we mentioned earlier. So that's just a summary. So this is also talking about how a pathway or moves. Okay, second order neuron are in the nucleus of the tractus solitarius. Axon, the second order neurons, run from the medial lemniscus and terminate in the postero-ventral nucleus of the thalamus, like we talked about. The third neuron, okay, third order neuron. The third order neuron are in the postero-ventral nucleus of the thalamus. Axons from the third order neuron project into the parietal lobe of the cerebral cortex. Taste center. Center for taste sensation is the orbicular insular cortex. Don't forget. Orbicular insular cortex, that's where we have taste sensation, okay? Those are the taste centers. In the lower part of the central gyrus, which receives cutaneous sensation from the face, thus the taste fibers do not have an independent cortical projection. They rely on other fibers. Okay, so this is just the same thing talking about how the taste pathway is able to move. So you guys go and pay attention on this. Read books like Guyton, Gano, try to depict and capture how these are able to move. And other things that I've left out, you can be able to do them by yourself. So this actually concludes the pathway for gustation or test from the tongue up to centers for gustation. Yes, there's an image on the insular cortex. Yeah, same thing. So this is just showing summary of the 12 cranial nerves. You can be able to go through in your own time. I'm sure anatomy has done already a good job. So now, let's go to now the primary taste sensations. Okay, so these primary taste sensations are 5, like I said in the preamble. So the primary or fundamental taste sensations are divided into 5 types. There's 1, sweet, salt, sour, bitter, and umami. Okay, these are the 5. The one umami was recently discovered, so but we'll talk about it as we go on. So now, man can perceive more than 100 different tastes. Okay, and this is just a combination of the 5 basic taste sensations. Combination of taste sensation with other sensations. Sometimes taste sensation combines with other sensations to give rise to a different sensation. For example, combination of taste, smell, and touch senses give a sensation known as sensation of flavor. Combination of taste with pain gives a sensation of what we call ginger. Okay, so it's just a combination. So your brain is able to combine these things and come up with the appropriate taste sensation. So now, discrimination of different taste sensation. Earlier, it was believed that different areas of the tongue were specialized for different taste sensation. Now it is clear that all areas of the tongue give response to all types of taste sensation. Remember, when you're in high school, that's what you are being taught. But all areas of the tongue are able to depict these different types of taste sensation. Usually, in low concentration of taste substance, each taste bud gives response to one primary taste stimulus. However, in high concentration, the taste buds give response to more than one type of taste stimuli. It's also clear that each afferent nerve fiber from the taste buds carry impulses of one taste sensation. Taste sensation and chemical constitution. What makes up these chemicals? Okay, so now substances causing sour or salt taste are mostly electrolytes. Bitter and sweet tastes are caused by electrolyte or non-electrolytes. Okay, let me read again. Substances causing sour or salt tastes are mostly electrolytes. Bitter and sweet tastes are caused by electrolytes or non-electrolytes. Sweet taste. Sweet taste is produced mainly by organic substances like monosaccharides, polysaccharides, glycerol, alcohol, aldehydes, ketones, chloroform, inorganic substances which produce sweet taste. And just are lead and beryllium, sorry. Now, salty test. Salt test is produced by chloride of sodium, potassium, ammonia, nitrates. So these are the different types of salts that are able to produce the salty test. Okay. Sour test, sour is produced by hydrogen ions in acid and acids. Bitter taste is produced by inorganic substances like guanine, trypsin, morphine, glucosides, etc., etc. Umami is the most recently recognized taste sensation. Umami is a Japanese word meaning delicious. Receptors of this taste sensation respond to glutamate. Okay, that's what they respond to. So these are the different types of taste and thresholds that you are able to pick up if you are able to test something. Let's now go to taste transduction. We have already talked about the pathway, how they are connected, how from the tongue up to the higher centers, from the tongue up to the center for taste or gustations, for gustation. Now let's look at taste transduction, the mechanism of taste. So test transduction is the process by which test receptors converts chemical energy into action potentials in the test nerve fiber, which is correct, because basically this has to do with converting from chemical to electrical energy, because your brain can only perceive action potentials as information. So the chemicals that you ingested should be, or the chemicals that your cells, the receptors in your tongue or in your mouth or areas that are able to test, if they are able to pick up those signals, they're in a chemical nature. They have to be converted into what we call electrical or action potentials. So now receptors of taste sensation are chemoreceptors, which are stimulated by substances dissolved in the mouth by saliva. The dissolved substances act on the microvilli of the taste receptors exposed in the taste pouch, causes the development of the receptor potential in the receptor cells. So basically, like we were talking about olfaction, if you remember, mucus is able to dissolve the chemicals. In this episode or in this topic, we are talking about saliva, which is able to also do the needful, okay? Dissolving the chemicals so that it can be picked up by these test buds. So this in turn is responsible for the generation of action potential in the sensory neuron. Test receptors, generally, test receptor is a G protein coupled receptor, GPCR. Okay? G protein coupled receptor. It's also called G protein gasdusine. G protein gasdusine. So here's just a summary which shows how these different types of tastes are picked up and how they are depolarized or they are converted to what we call action potentials. So here you can be able to see there's bitter, okay, there's sweet, umami, there's sour, and salt. So you can find that here at bitter, as you can remember, what constitutes of a bitter taste. So we are going to have, it binds, those chemicals are going to bind the G protein coupled membrane receptor. Remember, GPCR, yeah. So they're going to bind there. After they bind there, we are going to have an increase in, triphosphate and calcium ions. Once this increases, it's going to cause opening up of the therapy channels, which are going to cause an influx and cause what we call polarization. When you go on sweet or umami, binds G protein coupled membrane receptors, causing an increase in IP3 and calcium, opening up the TRP channels, and then causing what we call depolarization. Sour, as you know, the hydrogen ions were mentioned, enters through the membrane sodium channels, ENAC, then causing what we call depolarization also, okay? Because an action potential is initiated by what we call depolarization. So there's depolarization and there is repolarization, okay? Then as you go on, you're going to realize that enters through membrane sodium channels and it's going to cause also another depolarization. And this is the end of the session.